So TG-1801 is, as you've described, a bispecific antibody, but in contrast to many of the other bispecific antibodies which are currently under development, which target CD20 and CD3, in the case of B-cell lymphoma, the mechanism of action of TG-1801 is slightly different. As you've alluded to, CD47 is one of the specificities for this molecule, and CD47 sends a don't eat me signal to macrophages, which are part of the body's mechanism for recognition of foreign antigens, including tumours. So the inhibition of CD47 is not entirely novel in lymphoma or in hematologic malignancy. There is an approved compound called magrolimab, which targets CD47, and this works by blocking that don't eat me signal and inducing macrophage phagocytosis of malignant cells. And that is being explored in myelodysplastic syndrome and has been evaluated in B-cell lymphoma as well. The difference between TG-1801 and, for instance, magrolimab is the bispecificity for CD19. The interaction with CD19 in this case, rather than being a T-cell engager, because it's not, actually it's because CD19 gives the antibody specificity. We see less off-target. Less off-target toxicity by confining the effectiveness of the molecule to cells expressing CD19 only. And the reason that this is relevant is because one of the problems in patients treated with magrolimab, which is purely a CD47 antibody, is that patients get hematologic toxicity, in particular anemia, because CD47 is widely expressed on erythrocytes, and so you get off-target destruction of red blood cells. So in this first-in-human study, we performed first dose escalation in monotherapy and then expansion into a combination study with Ublituximab, which is a type 2 glycoengineered CD20 monoclonal antibody, which has greater affinity and causes greater ADCC than rituximab. And we saw, firstly, let me speak about the safety. It was a relatively small study, 30 patients, 14 treated with monotherapy and the remainder treated on the combination arms. In the monotherapy cohorts, we saw that there was reasonable activity, more in indolent lymphoma subtypes such as follicular and marginal zone lymphoma, with a few responses there, less activity in diffuse large B-cell lymphoma. But when you combine the agent with Ublituximab, so TG-1801 and Ublituximab together, we started to see a stronger signal for efficacy, and the waterfall plots clearly show greater reduction in tumour burden for both aggressive lymphomas such as diffuse large B-cell lymphoma, as well as the indolent lymphoma subtypes. The treatment, both as monotherapy and in combination, is well tolerated and has very few adverse events. It's given as an intravenous infusion, as most monoclonal antibodies are. The toxicities are mostly low grade, and we did not observe any dose-limiting toxicity. And many of these patients have had durable responses, even going out past 2 years, which for a non-toxic antibody treatment for someone with relapsed large cell lymphoma is certainly useful.